Thank you. Good afternoon, everyone. So I'm Semira Nusslé. I'm the co-founder and chief medical officer of Genome, the Swiss epigenetics lab. So we'll dig a bit into epigenetics in this talk. Lifestyle, stress, environment can all modify gene expression, thus health span, thus longevity, of course, through epigenetic processes. I'm not teaching you anything new here, I'm sure, but let's dig a bit into these epigenetic processes. So here we have the representation of DNA. It's a double helix made of 4 bases, 8 TCGs, and you can see that the Cs, the cytosines, when we talk about DNA methylation here, they can have the addition of one methyl group, CH3. And when there's this addition, it will completely change the 3D structure of this base. And this has the consequence that it will influence how much DNA is wrapped around itself, thus inactive, or open and active. Because when the DNA is open, the genes can be expressed because the enzymes, the transcription machinery, RNA polymerase can bind to the DNA and express the genes. So that's one of the epigenetic processes that is called DNA methylation. And what's really interesting is that we know now with lots of evidence that different factors can influence the presence or absence of these methyl groups, such as development. Of course, during development, some genes are switched on and others are switched off. And also environmental factors like lifestyle, environmental exposures, and illnesses can influence this landscape of DNA methylation. So let's focus now on one of the significant external factors that influence health and these DNA methylation processes. It's chronic stress. It's known since a long time in epidemiology that chronic stress is a risk factor for mortality, for aging, and for chronic diseases, and of course for mental health. How does it work? When there's a stress, we have a coordinated response that is involving hormones, stress hormones, and neurological pathways. It's called a neuroendocrine response. This involves the HPA axis with the expression of with the secretion of hormones like cortisol, and also the central nervous system, as well as the autonomous system. And the goal of this response is to influence all of organs so they are ready to fight and flight, to react to the stress trigger. So that's, most of the time, that's a desired reaction, right? We should fight or flight sometimes. The problem happens when the triggers, the stress, lasts for a longer period. Then the subtle changes that happen to the organs due to this neuroendocrine response will make them start to malfunction. And this is the beginning of chronic diseases. So this concept was studied since a long time. It was also measured mainly in epidemiological studies under the name of allostatic load. Allostatic load is a way of measuring this chronic stress impact, and historically was done by measuring a large panel of blood parameters encompassing markers of chronic inflammation, markers of metabolic health, of stress hormones, clinical parameters, as well as anthropomorphic measurements. And this allostatic load concept was studied and showed the importance of chronic stress as being a risk factor, like I said before, for aging, mortality, and these chronic diseases. So Because it's really important to tackle that, we've developed a new program to support healthcare professionals to help their patients who have stress-related complaints. And that's a program that is based on epigenetic markers of stress, as well as the designing of a personalized approach to make this stress impact decrease, and that is reflected into the epigenetic markers. So how does it work? So first, we assess patients' eligibility with a questionnaire, and then there's a blood collection, because we can measure the epigenetic markers from white blood cells. And then the sample is shipped to a lab in Switzerland. We are a spin-off of the Lausanne University Hospital. We are located near Lake Geneva on the Biopole campus, which is a life science campus. And our lab is authorized for epigenetic analysis, and we store all data in Switzerland. Everything is done in our lab. To measure that, it's a 4-day pipeline of analysis that required qualified personnel to analyze DNA methylation with dedicated technique and equipment. The technique that we use is microarray sequencing of bisulfite-treated DNA. Bisulfite is a chemical agent that will reveal the methylations. And we're a small lab, but we have a high capacity to perform hundreds of tests per week. We specialized in giving very fast turnaround times. That's it. So what is measured exactly? We've developed this epigenetic signature of chronic stress. There are 32 epigenetic markers that are located in the genes here and are associated with chronic stress. And the genes encompass different physiological systems that are impacted by stress, like the metabolic system, cardiovascular system, immune system, and neuroendocrine system. This signature is patent pending, publication pending, and to our knowledge, we don't have no known direct competition. How was it developed? So it's an epigenetic signature that reflects the concept of allostatic load. So we don't need to measure all this large panel of markers, clinical and blood markers, but we can concentrate on only measuring the DNA methylation markers. This was developed together with the University Center Unisanté in Lausanne, Switzerland, on the Swiss population as a reference in a Swiss population-based study called KIPOG. And it was validated on a Scottish cohort, Lothian birth cohort. And what we see here is that the epigenetic signature of allostatic load is very well correlated and significantly correlated with the clinical signature of allostatic load. And what's interesting also on the right-hand side is that we see that the odds ratio for the association between allostatic load and a history of cardiovascular disease is actually higher and significant and higher for the epigenetic signature more than just the clinical signature of allostatic load. So then today what we have developed is a report, is a clinical report where we translate the information from these genes into understandable and explainable measurement. We dose allostatic load on a scale from 0 to 4, and then we give an insight because we measure the genes that have functions in these different systems. We can give an insight. Is stress impacting already for this particular patient more the cardiovascular system or the metabolic system? And this gives some guidance for the professionals to design a personalized program for the patients that is based on these epigenetic results. And the program have the aim to reduce epigenetic impact and this can be followed over time because these epigenetic markers are dynamic and they will change. So that's us. We are a small team of 6. We founded the company in 2019. We've been already serving clients for 4 years now. Very happy to have among them the very prestigious longevity clinic, namely in Switzerland, and we were supported by a lot of grants and awards in our ecosystem. And that's it. Thank you very much for your attention, and please come see us at our booth.
Yeah, thanks so much, Semira. That was lovely. Yes, as Semira mentioned, they have a booth outside. In fact, I should take this opportunity to mention that all of our sponsors, I think every single one of them has a booth of some kind outside. And also, of course, we have poster presenters in the room upstairs where we'll have food and drinks after the session ends. So don't go anywhere yet. Are there any questions for Semira? Microphones, where are we? Who's got a microphone already? Anyone? Here we go. I'll do. Yeah, we'll start with that. Seems to be lots of questions.
That was a really good talk. Thank you very much. I was just curious on the bisulfate treatment, which can be quite harsh to the DNA. Have you considered techniques like nanopore sequencing, for example, which is just measuring the DNA itself?
Yeah, thank you. It's true that bisulfite is a difficult treatment, and it requires good quality of DNA, and it's right. I agree with you. So we are looking forward very much to use nanopore. So if you have any information on the availability and also the accuracy, I would be extremely interested, because last time I checked, we were not completely there yet, but I might not be up to date. So happy to chat.
Hi, Semira. Thank you for your speech. It's very interesting to me. I would like to ask you about the treatments that you said, according to immunity, according to stress, balance, stress, everything. If you are sending them for any intermittent fasting or any nootropics, if you use that in your treatments, or I would like to understand more about the examples that you do in your treatments for lifestyle and improving their lives, like if you believe about intermittent fasting or any fasting. Okay, thank you.
Thank you very much for your question. Yes, it's maybe important that I precise that we do not sell treatments. We really sell a test, and not really the treatments. We are happy to provide with advice, recommendations, but the treatments are in the hands of the healthcare professionals that will use that with our patients. We can give, what we have is a portfolio of evidence-based dietary supplementation, for example, or lifestyle interventions that we can recommend. We're more a lab. Yes?
Was there a broad ethnic diversity in the cohorts you used for your studies?
Yeah, that's a good question. It's always a problem. So for this one, it's unfortunately we don't have a lot of diversity. We had to do with the epidemiological data that were available, because we rely on large sample sizes, and we get the data that we get. So funding people who fund research, please fund more diverse studies. Thank you.
All right, thank you.
Thank you.